Hello, my name is Maddy, and today we're going to talk about expansion microscopy. For centuries, researchers have been trying to develop ways to visualize small biological systems by magnifying their image. Since the first microscope, we have made huge progress. We have developed light microscopes and electron microscopes to accomplish magnifying images and achieving nanoscale resolution of biological samples. These microscopes we have designed, while very useful for a number of biological studies, are limited by either the resolutions they can achieve or the samples that they can image. For instance, light microscopes can generally collect images of samples with a resolution of 200 to 300 nanometers. This means they can only distinguish between two parts of a sample as long as they are 200 nanometers apart or more. The strongest electron microscope can capture images with a resolution of 0.05 nanometers, which means they can detect two parts as separate as long as they are 0.05 nanometers apart or more. This is wonderful, but these electron microscopes need the samples they image to be very tiny so electrons can pass through them. This means larger biological systems can't be imaged using these microscopes. Also, electron microscopes are very expensive, and scientists don't usually enjoy very expensive things. So if we want to see a biological system too big to view with an electron microscope, but at a much greater resolution than a light microscope, what can we do? A team of scientists at the Massachusetts Institute of Technology wondered, what if instead of trying to develop a new microscope to see our original samples at higher resolutions, we started off with an expanded version of our original samples and used the microscopes we have on those expanded samples to achieve super resolution. They developed a technique that uses a polymer found in diapers, sodium polyacrylate, which can expand when exposed to water. These polymers can be arranged to form a matrix. The goal of expansion microscopy is to incorporate your biological sample into this matrix so that it can expand with it. In order to attach the sample to the polymer matrix, small anchoring molecules are introduced which bind to the regions of the sample and then anchor themselves to the polymers. Different anchoring molecules can additionally act as labeling probes and have been developed for different types of samples, such as proteins, cell membranes, and RNA. The anchoring molecules you use thus depends on the samples you want to study and what structures you want to identify. After the anchoring proteins have tethered your sample to the matrix, your sample needs to be digested so that it can break apart and expand with the polymers. To accomplish digestion, you can add either an enzyme, such as proteinase K, or apply an increased temperature and detergent treatment. Now the sample has been secured in the matrix and digested, so once water is added, it should expand isotropically with the polymer matrix. This means that it will move in every direction at an equal rate, and that you will get a resulting expanded sample directly proportional to your original. Samples can expand up to 4.5 times its original size. This means a microscope that originally achieved a resolution of 300 nanometers can now view your sample with a resolution of approximately 70 nanometers. Depending on your sample and what you want to accomplish in your study, you can also apply fluorescent antibodies before or after you expand your samples. You can help identify specific characteristics of your sample at the much higher resolution expansion microscopy offers. Here are examples of a nucleus after being expanded, compared to an unexpanded nucleus treated with fluorescent antibodies in a study done by Boutner et al. In 2021. With this technique, scientists in various fields have been able to study samples ranging from very small systems, such as synapses between neurons, to whole organisms, such as C. elegans. In the end, expansion microscopy is a helpful super-resolution technique that can help scientists better understand biological systems. While expansion microscopy cannot be used on live cells and requires optimization of protocol with each sample used, it is a cost-effective and precise way to achieve high-resolution images. Thank you. If you need expansion on any of the expansion microscopy concepts, please feel free to visit these websites. Hope you have a wonderful day.